Musk-induced signaling cascade causing nicotinic acetylcholine receptor clustering. Dok7 is an adaptor protein in stabilizing and activating musk signaling, inducing musk dimerization, which activates rhabdin, a scaffold protein. Thin filaments slide across one another, which makes the entire sarcomere unit contract, which contracts the myofibrils and the entire muscle fiber, with phosphocreatine producing ATP as well as AMP. Different mechanisms of action. Smooth muscle, that governs subconscious automated actions lining internal tracts. Cardiac muscle, that operates in a similar process, however contains striations from filamental fibers, yet around the heart. And finally skeletal muscle, which facilitates movement and motion. Skeletal muscle is made up of large bundles of long cells, which is known as muscle fibers. These muscle fibers contain a cell membrane, known as the sarcolemma. This foldable membrane penetrates deep into the muscle fibers. Transverse T-tubules to allow the depolarizations to reach all throughout the muscle and induce calcium release for muscle contractions. T-tubules are present along the Z lines of the sarcomere in cardiac and on the M lines in skeletal muscle. These membranes further move through the sarcoplasm within muscles, and these inner foldings of membranes associated with another tubular network of the sarcoplasmic reticulum, primarily functioning to store calcium ions, forming what are known as triads in skeletal muscle, with two sarcoplasmic reticulum membranes associating with each T-tubule, and dyads in cardiac muscle with one sarcoplasmic reticulum membrane. The individual components of these muscle fibers are myofibrils that create this complex structure. As a deeper dive into the structure of these cells, these myofibrils contain thick and thin filaments. These thick filaments are known as myosin filaments, as they are built on the protein myosin, as well as the thin filaments consisting of actin. This combination of actin and myosin within skeletal muscle cells creates the fundamental unit of these myofibrils, which are known as the sarcomeres. They consist of an A band containing only thin actin filaments, often diagrammed in blue, an I band at its core housing myosin filaments with a thin interleaving layer of actin filaments, and a H zone. Now here is how the muscle contraction is governed. The Z line marks the outer component of the sarcomere, to which the large protein titin and nebulin anchor myosin and also contribute to elastic properties of the muscle. Muscle contraction is governed by the sliding filament theory, where the filaments themselves do not contract. Thick and thin filaments slide across one another, which makes the entire sarcomere unit contract, which contracts the myofibrils and the entire muscle fiber. The Z lines move inwards, while the A bands remain completely the same in length. The I bands constrict as well as the H zone in order to create a shorter sarcomere that contracts, which is what generates muscle contraction. Within the sarcomere, myosin slides along actin to contract the muscle fibers using ATP, bearing in mind that each skeletal muscle fiber is a single cylindrical muscle cell. Skeletal muscle is composed of many cells known as muscle fibers that run distantly, as well as being multinucleate and containing long organelles known as myofibrils that hold the mechanism for muscle contraction within, the fundamental unit of the sarcomere. Muscles attach onto bones by tendons, while ligaments join bones together. Muscles work to move the bone, where two pairs of muscles known as the agonist and antagonist control a key function in the body, such as flexion of the knee by the quadricep, opposed by the hamstrings. Myosin contains a globular head that is able to bind to actin filaments that contain actin-myosin binding sites. At the muscle's resting potential, these binding sites are occupied by another protein known as tropomyosin, serving to hold these thin actin filaments together. Along the length of the tropomyosin there is another protein troponin bound, with each troponin having multiple subunits associating with the different interactions it makes. For example, troponin C is what contains the calcium binding sites, with 4 in skeletal muscle and 3 in cardiac. There is also a separate binding site for ATP for catalyzing and powering the process of respiration for muscle contraction. Now we arrive at the neuromuscular junction, the last activation pathway before an effector, a muscle, can bring about a response. An action potential arriving at the axon terminal, the depolarizing wave, opens L-type calcium channels, with the calcium influx causing vesicular fusion and exocytosis of acetylcholine. Here is where the CAT's property of quantum neurotransmitter release occurs and produces the micro end-play postsynaptic potentials, or EPSPs, that, though don't reach threshold, act to cause minute and micro depolarizations. Here we have a motor neuron releasing acetylcholine across a cholinergic synaptic cleft. Acetylcholine diffuses across the synaptic cleft to bind to nicotinic acetylcholine receptors. Here, there is a very intricate mechanism to achieve motor end-play depolarization. Signaling cascade causing nicotinic acetylcholine receptor clustering. Dok7 is an adapter protein in stabilizing and activating MuSK signaling, inducing MuSK dimerization, which activates rhabdin, a scaffold protein. On a clinical standpoint, there can be multiple causes of myasthenia gravis, which is an autoimmune response and can cause antibodies to attack any of these proteins and players in this intricate mechanism, causing more rare forms of the disease than just the immune response against the nicotinic acetylcholine receptors. The postsynaptic membrane of the motor end plate functions similarly to that of other membranes, yet these junctions have two key differences. Firstly, they contain a far greater number of receptors in order to trigger greater action potential to bring about strong effects, contraction of muscles. Secondly, this membrane has a lot of folds that form clefts, in which the enzyme acetylcholinesterase is stored in order to break down acetylcholine that does not bind to these nicotinic acetylcholine receptors, terminating signaling. Acetylcholine is always excitatory, as well as neuromuscular junctions, which is not the case in other parasympathetic neurons, which is usually inhibitory, for example at the heart with muscarinic receptors. Drugs can also have significant effects at synapses, for example amphetamines, which trigger the release of dopamine as a neurotransmitter, despite its presence as a hormone as well, accentuating the effects of the dopamine, namely increasing alertness. Calcium ion channels being blocked through drugs such as opioids also result in fewer vesicles fusing to membranes, resulting in muscle weakness and paralysis. This is how muscle contraction communicates with the nervous system. Botulinum toxin is competitively binding to nicotinic acetylcholine receptors and is also utilized as an anesthetic, causing paralysis, while sarin gas, on the other hand, is a suicide inhibitor of acetylcholinesterase. When an action potential arrives at the neuromuscular junction, acetylcholine, an excitatory neurotransmitter for muscle contractions, binds to many nicotinic cholinergic receptors. At cholinergic synapses, the arrival of an action potential stimulates voltage-gated calcium ion channels in the presynaptic neuron to open, with calcium ions diffusing into the synaptic cleft. The influx of calcium ions causes the vesicles to fuse with the presynaptic membrane, and they release a neurotransmitter, acetylcholine, into the synaptic cleft by exocytosis. Acetylcholine diffuses across the synaptic cleft, binding to cholinergic receptors on the postsynaptic membrane. Nicotine acetylcholine receptors are ionotropic and aren't very selectively permeable, so they allow cation influx, with the influx of sodium ions creating a difference in voltage, triggering an action potential or not, which can be denoted in a binary form at the neuromuscular junction as a 0 or a 1. The muscle either contracts or relaxes, with lengthening playing an antagonistic role at an opposing muscle contraction. When an action potential arrives from a motor neuron to stimulate a muscle cell, it depolarizes the sarcolemma. Depolarization spreads down the transverse T-tubules, which is detected by DHPR receptors, which act as a voltage sensor but are really a calcium channel. DHPRs are coupled to ryanodine receptors on the sarcoplasmic reticulum, triggering the release of stored calcium ions with the influx of calcium into the cell, which binds to troponin to initiate muscular contraction through the cross-bridge cycle. The calcium release is crucial for muscle contraction, which contributes to the phenomenon of tetany, where the summation of action potentials stimulate calcium release, which provides a sustained contraction. Muscle contraction can be depicted as a simplistic model of a head and a receptor. Calcium ions will bind to a protein known as tropomyosin to change its shape and unveil the actin filament binding site that slides over the myosin filaments containing globular heads that serve to attach and detach.
forming many actin-myosin cross-bridge cycles in a sliding mechanism contractile process, as an eccentric lengthening muscle contraction occurs, where a force applied to the muscle exceeds the momentary force produced by the muscle itself, resulting in the forced lengthening of the muscle tension system while contracting. Most ATP is generated by phosphorylation for this process, and is a process that succeeds off the back of aerobic respiration. In anaerobic respiration, ATP is made rapidly by glycolysis, the breakdown of glucose, which results in the production of pyruvate, and in an anaerobic state forms lactate, building up in the muscle cells as lactic acid and having toxic effects which enforce muscular,
with phosphocreatine producing ATP as well as AMP.
Energy is released slowly through aerobic respiration, and these fibers have developed blood supply mechanisms with great quantities of oxygenated blood arriving to them, as well as the protein myoglobin, which binds oxygen effectively for the release of energy, with its reddish colour giving these muscle fibers a darker red appearance. They also have many mitochondria. In contrast, fast-twitch muscle fibers have less mitochondria, myoglobin and blood vessels, relying on anaerobic respiration processes involving glycolysis, as well as harnessing the sheer might of the ATP phosphocreatine system, contributing to our different strengths and attributes that adds to our uniqueness as a species.
Now, onto cardiac and smooth muscle, which have a completely different form of action, potential propagation to skeletal muscle. Smooth muscle, first of all, doesn't have striations, but instead fibrils are attached to dense bodies, and cells are joined by gap junctions to propagate electrical impulses via electrical synapses directly between cells. Smooth muscle contractions are slower but sustained to ensure movement along a pressure gradient, and does not require neuronal activity or any voltage-gated channels to generate tension. In a similar way, cardiac muscle, although has striations, it forms a functional syncytium with intercalated discs connecting myocytes, in which electrical impulses spread throughout the heart to effectively and regularly.
Smooth muscle depolarization occurs with calcium influx after threshold is reached by the ANS hormones through receptor-operated channels, or stretch through stretch-activated ion channels. Calcium binds to calmodulin to form a calcium-calmodulin complex. Calcium-calmodulin activates a myosin light chain kinase, which results in myosin light chains being phosphorylated, which increases myosin activity and binding to actin, resulting in the contraction.
Termination is through myosin light chain phosphatase, which removes the phosphate. However, if myosin light chains are dephosphorylated with thin and thick filaments bound, then the latch bridge is formed, which maintains tension without ATP consumption or cross-bridge cycling. Finally, cardiac muscle and its differences to the skeletal muscle. Cardiac circa has an inhibitor called phospholamban. Cardiac L-type calcium voltage-gated channels conduct calcium. This stimulates release from RyR1 2 receptors, whereas in skeletal muscle, DHPR receptors, which are equivalent to the L-type calcium voltage-gated channels, are mechanically coupled to RyR1 1 receptors. Calcium is released per action potential in the skeletal muscle is always in excess to fully activate the fiber. For cardiac muscle, it is always non-supramaximal sarcoplasmic reticular calcium release per action potential, which allows you to modulate the strength by changing the influx of calcium. This can be shown through the Bowditch effect, where an increased heart rate is formed from increased sodium overload of the sodium-potassium ATPase being overwhelmed and having a lower driving force for the NCX channel to work, which means that more calcium accumulates within the cardiac myocyte and increased strength, and more calcium ends up in the sarcoplasmic reticulum via circa.
Thank you for tuning in on muscle fibers and really looking at them at a deeper level. You could even say the quantum level. Please like and subscribe if you want to get any more deep and broad insights into any fields in medicine and science.